So let's begin with transport across membranes. We were talking about how important it is for things to move in and out of cells. Whoops, I can't seem to spell for some reason here. Moving in and out of cells. So we've talked about the fluid mosaic model of the membrane. And what I'll do is I'll just draw a very quick sort of picture of it. Okay, these are the little circles. I'm drawing them very fast. This is not meant to be accurate diagrammatically. It's just meant to give us something to refer to as we talk. So in here we have the little tails, right, of the molecules. And what I want to show here is one of those integral proteins, the big tangled up string-like molecule of the protein in the middle. And we're going to look at all the different ways that you can get through a membrane. In order to do that, we have to review a couple of concepts. We have to remember words like diffusion and osmosis. Right? Are those words that are sort of familiar to you in some way? Okay, so let's go through that, first of all. So we'll come back to that picture. I'll bounce back up and down there later. Let's talk about these two concepts, diffusion and osmosis, to understand those first. So diffusion is basically the movement from what we call a high concentration to a low concentration. But now that we're seniors, we're going to use some better words to describe that. Concentration. So let's think about what that actually means by using an analogy, another thing that we're familiar with that can be thought of in a similar way. So let's talk about, move up here, a bit of jumping around, but what this is essentially called when we have high and low concentrations is something called a concentration gradient. So we want to use the word gradient and understand what the word gradient means. So to do that, I'm going to have you imagine some level ground. And if we take a shovel and we start digging holes on this side, and actually let's dig holes on the other side. Not that it really matters, but we're going to dig a hole on this side with our shovel, okay? And so there's our hole, and we're going to pile that dirt up over here. So we keep digging holes on this side, so we end up getting a bigger and a bigger hole on this side. And on the other side, we keep piling up the dirt. Well, what we're essentially doing is we're creating a hill. If we keep doing this over time, and then we smooth it all out, we'll basically make a hill. Where we used to have the even ground like this, we now have a high spot where there's more dirt and a low spot where there's a hole. Pretty straightforward, right? Well, this is called a gradient because we've taken something that was flat and we've made one side higher and one side lower. This would be called a gradient of height. So a height gradient right? Or I guess you could call it a dirt gradient. Maybe in brackets we could put it's a dirt gradient because it refers to how much dirt is in one place as opposed to in another. Essentially, it's a hill. That's what we use the word hill to mean. A gradient is a hill. Or sometimes we use the word grade, which comes from the word gradient. When truckers see the sign on the road that says a steep grade ahead, it means a hill. So that's very easy for us to understand. Well, the thing is we can mathematically describe a hill, and you've learned to do this in grade 9 math already, even though you don't realize it. You've learned to describe hills by using mathematical equations, and of course, the key part of the equations is the slope. Another word for hill is a slope, right? So a gradient could also be thought of as a slope or a hill, and you can use mathematics to describe mathematical gradients and slopes for anything. It doesn't have to be dirt. Right? We could pile up anything on one side and less of it on the other and create a gradient. So if we talk about what's called a concentration gradient, it's the same idea. A concentration gradient is a hill of concentration. Well, what does that mean? Well, if we take a space and we put lots of something on one side, pretend these are molecules dissolved in the water, and we have less of them over here, well, we could model that as a hill. Okay, we could say, well, that's just like drawing a hill, where the high side is here and the low side is over here. It's a gradient from high to low. And we're talking not about actual dirt anymore, we're just talking about what's high or what's low is where the molecules are. There's more molecules on the left, it's the high side. And then, of course, what happens on hills? If you put a ball on a hill, it automatically rolls down. It doesn't need a push, doesn't need help, right? It'll just roll. And so what happens with gradients is that some things will move along the gradient just like a ball rolling down a hill. In this case, what will happen is the high side, the molecules will move from the high side to the low side. They're kind of rolling down the gradient towards where the low is, just like a ball on a hill. You can have a gradient of concentration. You can have a gradient of electric charge, which we call voltage. There's all kinds of different gradients you could make. But we're specifically going to look at concentration gradients right now. So we have to imagine, if this was the case, if we had all these little dots representing molecules in a solution, what they would do is wherever it was high, they would move towards where it was low, automatically, without being pushed or helped. And then what would happen is they would keep going until the gradient was gone. Well, if the gradient's gone, that means that they've all moved so that it's evenly spread all over the place. And now there's no place that's higher or lower, so there's no more hill. And the movement stops. Now, actually, the movement doesn't stop. This is kind of interesting. These molecules are constantly shifting in their place. And so what happens is some of them are moving this way, some are moving this way, some are moving that way. But if you add up all the movements, it basically adds up to nothing. They cancel each other out. This is what's called a dynamic equilibrium. And it's a really interesting thing, but you don't think about this until you actually think about it. You don't see the difference. In a room like this, we could have boys and girls all just standing all around, boys and girls. So I'll put some boys in, and then I'll put blue dots for the girls. And if they're all just standing still, that's what we would call a static equilibrium. A static equilibrium means that nothing is really moving, and that's everything is nicely even spread out. There's no boys on one side or girls on the other. But a dynamic equilibrium is, it looks almost the same. What you have here are boys and girls, but they're not standing still. They're all milling about the room, moving. So there's constant swirling and moving of the boys and constant swirling and moving of the girls. But if you were to take a snapshot with a camera at any moment, it would just be even spread out all over the place. The constant movement is not producing any one spot where there's more girls or boys. It's just constant milling about. That's always pretty much balanced. That's what dynamic equilibrium means. And most equilibriums in real life are these kinds of dynamic equilibriums, especially with molecules, because molecules are always moving. They're always moving around. Okay, so we understand that a gradient can be made. The process of sliding down a gradient is called diffusion. So diffusion is simply things sliding. Well, let's not put things. It's molecules or compounds or solutes. When we're talking about concentrations, the word we use is solute to describe the things that are inside the solution, right? Bits of sugar, bits of starch, bits of molecules, bits of it could even be dirt or mud or anything. It's is simply, it should be a Y, solute rolling down the gradient. which means it's always going from high to low. And so diffusion could be a way for things to get into the cell. So let's go back to our membrane. Well, instead of going back, you know what? I'll just draw it again because it's pretty simple. Here's our membrane. And we will not need a protein. So I'm just going to draw a part of the membrane that doesn't have any proteins near it, because the proteins won't be necessary for this kind of, and the tails I'll just do like this. There we go. There's our membrane. Okay. Now remember, these molecules are actually, they're not joined together by chemical bonds. They're joined together by what we call van der Waals forces, intermolecular forces. So I want to make sure that it doesn't look like they're all stuck together by something. So in my hurry, I drew it like a spiral. It should be. So what that means is that little things, small molecules, would be able to just sort of creep their way right between, like kind of like pushing your way through a crowd, right? I'm just going to erase up top because I need some more space here. So let's imagine we had oxygen gas. Oxygen gas, O2, is really just two oxygen molecules holding hands like this, right? It's a very small molecule. It's 2 atoms big. So it's tiny. And so what can happen is, if you had a place where the oxygen molecules were in high concentration, let's say here, and I'll just use little blue circles, right, to show all the different oxygen molecules. But here, there's only a few. Why would there be so many here? Well, maybe this is somewhere near the lungs. Maybe this membrane is a cell lining the lungs. And when you breathe in, out here inside the lung cavities, whoops, what happened there? Come back. Okay, we're still going. Every once in a while, weird stuff happens. I don't know why. The lung cavity, called the alveoli, right? When you breathe in, it goes into your tubes and down into your lungs. This could be that spot where you just took a breath of fresh air full of oxygen. But inside the cells, there's not very much oxygen. Maybe this is the bloodstream. Maybe these cells line the bloodstream. They line the vessels. And the oxygen has to get into the blood. So this cell, if it's a blood vessel that lines the walls of your blood vessels, the oxygen has to get through. Well, clearly there's a concentration gradient. There's a gradient here from high to low because we have more oxygen on one side than we have on the other. And that means that oxygen will want to diffuse. It will want to roll down the membrane. Roll down the gradient. And what that means is it'll squeeze its way right through the molecules, woo-hoo-hoo, right through there, and it will begin to move in this direction. This is diffusion. And it happens because oxygen is small enough to squeeze through the membrane all on its own. So the first and easiest way to get things into a cell, or out for that matter, is by simple diffusion. If you have more stuff on one side and it can squeeze through the membrane, it will diffuse, follow the gradient, and find its way out. So simple diffusion is essentially the first way of getting things in and out of the cell. But it only works for little small tiny molecules, or molecules that are soluble in fat. So simple diffusion only works for really small molecules. So these small molecules would be, for example, oxygen gas. Carbon dioxide is also pretty small, right? And then we'll talk about water later, because it's a whole different process, but water can move also pretty easily through a membrane, etc. So small things. Or lipid-soluble molecules. Lipid-soluble molecules are molecules that are essentially non-polar, and therefore they like to be inside fat. Remember the fat tails of the membrane are not polar. They're lipid molecules with no polarity in the tails. So to get through the membrane, if you like being inside, or if you can dissolve in the non-polar substance, then you could essentially, even if you're fairly big, you could wiggle your way in here and literally dissolve your way through the membrane. Because you can dissolve in this inner layer, which is non-polar. So that can happen too. That happens, one of the molecules that does this quite often in our bodies is steroid hormones, which are special hormone molecules made of lipids and fats. And they can penetrate membranes much easier. And we often use steroid drugs to mimic these hormones to treat things because they can penetrate the membranes easier. So simple diffusion means squeezing through the membrane by following the concentration gradient. Okay, that one is easy. Now, what happens if you're bigger? What if you're a molecule of, say, glucose? C6H12O6. That's a lot of atoms. And I think if you draw a picture of glucose, it sort of looks like a shape like this. And has little, like that. It's a big sort of hexagon shape. It's a large molecule. And there's no way that it would be able to squeeze through a membrane, even if there was a concentration gradient. So what happens if you have a membrane? Okay. And this time we're going to put a gate in there because we're going to need help. Here's our protein. Gateway, the integral protein. The sugar molecules are far too large. So let me draw some sugar molecules here. I was going to try to draw hexagons, but now I'm just drawing blobs because that's too much work. So there's my sugars, okay? Over here I have very few sugars. So I do have a concentration gradient, right? There is a gradient of high to low here, but diffusion won't work because these molecules just keep bumping into the membrane and can't get through. They're too big. So what can happen instead is the cell can use another molecule as a signaling molecule to land on this protein. Or sometimes the glucose itself will cause this protein to open a channel. Remember we talked about how that can open up a channel inside? A pathway, a hole through the middle. And this channel is big enough for these glucose molecules to slide through. So it's kind of like diffusion. It's still going from high to low, but it's requiring a little bit of help. It requires a little help because it can't fit on its own. So in other words, we have to open a gate to let it through. Okay? So you can imagine this just like a gate. If you think about a fence, right? Here's a fence. Maybe it's a wrought iron fence. Well, if you have a whole bunch of mice over here and not so many mice on the inside, the mice can run right through the fence. They can squeeze right between the bars, no problem. But if you have a whole bunch of dogs over here, they can't squeeze through the bars, so they have to wait for somebody to come along and open a gate. And once you open the gate, all these dogs on one side will push and shove and make their way in. So it's still diffusion. It's still high to low, going down the gradient, but it requires the aid or the help of opening a channel in the proteins of the membrane. So we call that facilitated. Facilitated, whoops, let me spell that right. There's just a C in there. I'll just start again. The word facilitate means to help, right? Or to make happen. So facilitated diffusion is another way that cells can get stuff to happen. Now, the problem is, how does the cell do this? Well, it has to open the gate. It requires something to happen to get that gate open, which means that facilitated diffusion might require some extra energy involved. Regular diffusion doesn't require anything. It just happens automatically like a ball rolls down a hill. But facilitated diffusion requires you to open up a channel. And opening up the channel may require you to do something extra, which is an energy input. So sometimes cells have to work. If you remember physics, work is changing your energy. So if you want to use energy to change the cell to open gates and close gates, guess what? You have to do some work, which means it costs you something. So cells will often use their energy to control what moves in and out of the cell. However, the amount of energy required here is quite small. And it's also possible that the sugar molecules themselves could cause that gate to open. So typically, this kind of facilitated diffusion, along with regular diffusion, is called something, we call them passive transport. And the reason we call them passive transport is because they don't really require any energy added. The small bit, the opening of the gate, maybe, but even then it could just be because of the big sugar molecules being so concentrated. So passive transport is essentially stuff that is moving down a concentration gradient from high to low and would roll anyway, just like a ball rolls down a hill. Now, the problem with cells is that sometimes they face the opposite. Sometimes they have to get stuff out, but it's like pushing up a hill. If you've ever had to roll a ball up a hill, you know that you have to continuously push it with extra force. You have to use your muscle energy to roll it up the hill. So imagine, for instance, if a cell wanted to, let's say there were waste products inside of a cell, but outside there was lots of waste also. But the cell still wants to get rid of those waste products. They're not helping the cell. They might even be toxic if they build up. So if I open little channels in my membrane, that's not going to help, because that's going to mean diffusion. Everything's going to come in because it's high outside and low inside. So that won't work. That's the opposite of what I want. How does a cell get these things to go out against the concentration gradient? Well, in that case, what the cell can do is, again, utilize those protein gates. So we'll draw a little gate here. There's our membrane. And we'll draw our little protein gate. This time we're going to have, we'll put some molecules outside, but we'll put lots of them. The concentration is high. And in here we just have a few. So we have a concentration gradient from high to low. But we do not want to just open up holes, because everything's going to flood into the cell. So what can happen is the cell can use energy. And it uses energy in a cool form. Energy, I don't know if you know this yet, but energy is stored in cells in a molecule called ATP, adenosine triphosphate. And ATP energy is kind of like the battery of cells. Cells are made. Come back to me. Come back to me. I don't know why it keeps doing this. It's something to do with the plug. There we go. Okay, we're back. They're like batteries, ATP molecules. Your mitochondria make them. They charge up the batteries, essentially. The batteries are like, think about rechargeable batteries. These batteries go to the mitochondria, they get charged up, and then they have extra energy. And you'll learn all about that in grade 12 bio. All the complicated biological processes and how that works. But then as they float around, they can deliver that energy to other places. So one of the things they can do is they can deliver their energy. Let me just put a green molecule here, ATP. And when it attaches to the protein, it opens a channel. Remember how we said that opening channels is caused by molecules that attach, and you have to push and pull on the gate to get it to open, right? To make a hole through that tangled web of coiled up protein molecule. Well, ATP is commonly used to do that. And the energy of the ATP is transferred to the protein by attaching The P stands for phosphate. So the phosphate comes off of the ATP and it attaches to the gate, which leaves a molecule called ADP, adenosine diphosphate. So phosphate is actually an energy transferring molecule. If you stick a phosphate onto the protein, it could open the gate. But not only open the gate, it doesn't just open it. What it does is it provides energy to grab onto one of these molecules of waste and cause it to funnel up through the channel. It literally pushes it out. And so this molecule is pushed by the power of the ATP molecule. The energy from that ATP is used to push this molecule against the concentration gradient. So molecules are pushed through gates, against the concentration gradient. That's important. They're rolling uphill against the gradient by using the energy stored in ATP molecules. Transferring of that phosphate allows the energy to be moved from one place to another. And I said in grade 12, you'll learn the very intricate details of how that energy is transferred. But for now, we just need to understand. The key thing here, though, is that the cells are using up energy to push something up a hill. And they can do this, but it costs them. This is called active transport. In the other two examples, very little energy or no energy was needed because all you had to do is open the gates and let everything flow, right? It'll all flow downhill on its own. But here we need energy constantly because we have to push this molecule all the way up through the gate and push it out to where it doesn't want to go, to the high spot. So that's another way that cells can get things inside, but it costs them energy. So in order for cells to do this, they're going to have to also have a way to gather energy, right? In other words, they have to eat constantly to replenish their energy so they can continue to function by moving things in and out of their cell membrane. All right, what happens if you have something really big, too big to even get through a gate? Let's look at this example. Here's a cell. It's a big one-celled protozoan. It's called an amoeba. And it's one cell big, but it's you have to use a microscope to see them. They're still only the size of a cell. But maybe there's a little tiny cell over here. Maybe there's a bacteria cell or a small little cell like that. But you can tell by the picture, it's way too big to squeeze through a gate. In fact, we've zoomed out so far, and we can't even see the detail in the membrane anymore. The blue line represents the bilayer, right? But it's too small to allow for this big chunk of green thing, this big bacterial cell, to slide through. So what the cell does, because it's a fluid membrane, the membrane can ooze and move and stretch and twist, right? So the cell is able to push the membrane around by using currents of liquid inside. And the what we call the cytoskeleton of the cell, which is made of little tubules that connect all the parts inside. And so in a series of pictures, what would happen is this. This amoeba would twist and sort of move itself to sort of surround the little germ or the little bacteria cell like that. And then a little while later, it would keep doing that until it had completely surrounded it. So something like this. Oops, I gotta fix that. And it made like a little bubble. It reached around almost like arms. In fact, they actually call these extensions pseudopods, which means fake feet. And they reach around and they would touch. Now, once they touch, think about those molecules, right? Think about what's actually inside those blue lines. The little molecules, right, of one membrane are now sort of touching the molecules of another membrane, like this, right here at this spot. Because these molecules are fluid and liquid, they can just kind of merge with each other and bloop and kind of combine and take on a whole new shape. They can combine into one. And so what would happen is at that spot, the membrane would just sort of shift and bubble around the gap like this, and it would leave a bubble inside the cell like that. So part of the membrane forms the outside of this little bubble. I can just undo that, right? Part of the membrane is the bubble, and the other part just seals off the gap. And now we have a bubble inside. This bubble is called a vacuole, right? And this vacuole now contains this large object. Now what's the cell going to do with it? Well, probably what it will do is it will now use either active transport or diffusion or facilitated diffusion. And inside this blue membrane, right? Oops, let's go blue. Inside the blue membrane, I'm going to draw this really fast. These are the heads. There's my blue membrane. So I'm zooming in on the vacuole so we can see the membrane. There's the membrane. There's the tails inside. Okay? And my little green dude is in here. Now that it's inside the cell, the actual cell is much bigger, right? This is all inside the bigger cell. So now what can happen is molecules that are used to digest and degrade and break down or metabolize this little bacterial cell can be brought into the vacuole by using the facilitated diffusion we were talking about before, or the regular diffusion, probably facilitated diffusion. And once they go in here, they can attack and break this down into smaller little bits. And then those smaller little bits could then be pulled out back into the cell to be used. And if there was crap that you didn't want, if there was stuff in there that the cell didn't want at all, the leftovers could then be brought back to the edge, right? Once you're finished digesting it and you've taken out what you want, then what the cell can do is move the vacuole back to the edge like this, and all that's left in there is some waste products. And it can reverse the process, and as the membrane merges, it can open back up again, like this, and the waste products can be squirted outside of the cell, kind of like the reverse of what just happened. This process is called phagocytosis, because phago means to eat, food. Phagocytosis. And it's a way of getting very large things inside of the cell. So those are basically the three ways that cells can utilize or can get things in and out of them, utilizing diffusion and concentration gradients, or pushing against concentration gradients with active transport, or this kind of a thing, which can bring very large things into a cell. Your white blood cells would work like this. So those are the ways that things can get in and out of cells. I was going to talk about your white blood cells, for instance. What they do is they use this phagocytosis approach to engulf a germ or a bacteria or a virus particle or something in your body. And then they digest it and kill it and spit out the rest. Okay, so that's basically how things move in and out of cells. And that's how we're going to see cells are able to obtain nutrients. We'll just call that one done, and tomorrow we'll continue on with the next thing.